Hello, good afternoon. Thank you to everyone for coming to the webinar of the SEBBM. Today we have the group of regulación metabólica y nutrición, and the coordinator is Josep Villena. He is from Vall Research Institute, and today he has a guest, Francisco, and Josep is going to tell us and introduce him better than what I could do. Thank you.
Hi, good afternoon, everybody. Welcome to this webinar of the metabolic regulation and nutrition group of the SEBBM. Today we have with us Francisco José Ortega. He's a researcher of the Girona Biomedical Research Institute. He joined the institute in 2006 and he did his PhD over there. He graduated in 2012 in experimental science and sustainability. In 2016, he was appointed as a principal investigator under the umbrella of the Paris program of the Generalitat of Catalonia. And then in 2020, he was awarded a Miquel Servet position. After obtaining his I3 certification, Francisco was appointed as a lead researcher of the Cellular and Translational Research Group in the same institute where he has been conducting his research on adipose tissue biology. Also, he's a member of the SEBBM. And today he's going to talk us about the research line he's conducting in his lab on adipose tissue biology, as I said, and the relation that it has with pathologies such as obesity and metabolic disease. So, Francisco, welcome to these webinars, and the podium is yours.
Thank you very much, Pep. Let's share this screen. Now you can see the presentation, this first slide. Yes. Okay, I would like to first thank the PEP and the rest of the organizers of this event to give me the opportunity of talk to you about this research topic, which is, as a matter of fact, the leitmotif of my career as such. You see, I started my research activity in 2006. Back then, the Girona Biomedical Research Institute was just a teeny little room in this massive hospital, Dr. Josep A of Girona. I would say that the dream of a bunch of medical doctors that wanted to do some research of quality based on their respective areas of expertise. Here in the picture, you can see two of these senior researchers, José Manuel Fernández Real and Wilfredo Ricard. A little bit after starting my research activity in this incipient research team, my bosses back then brought me to the basement of this hospital. In there, they kept under key minus 80 degrees freeze. Inside, they had as many as 300 tubes falcons of 50 ml full up to the top with human fat. They told me, this is adipose tissue we've been collecting for the last 2 years, and now we need you to figure out what to do with all this material. It was, as I said, the beginning of my career and also the seed of what we know today as Fat Bank, the first collection of human adipose tissue available for biomedical research in this country. Today, the Fat Bank takes in samples and clinical information coming from 5 hospitals of reference in Spain. This is just to say that I've been working with human adipose tissue for so long that I have developed kind of an obsession to understand the nature of this paradigmatic tissue. You see, this is an organ that's plastic and dynamic, complex in a sense, formed by a number of different types of cells that coexist in some sort of unorganized hierarchy. In this milieu, if there is a master of ceremonies, that would be the mature adipocyte, this huge cell that takes in the energy of the body in the form of triglycerides, in a lipid droplet in the form of triglycerides. The key word here would be the energy, you know. I used to say that if the body was a country, then the fat deposit, the adipose tissue, would be its bank. And you know what happens with countries when banks fail. The collapse comes short after. This is something we have seen in these transgenic rat models that are deficient for the synthesis of adipose tissue, and also in these patients that suffer from a very rare genetic disease that are called laminopathies, in which they have not the ability of synthesize any adipose tissue. In these animal models and in these patients, the lack of adipose tissue promotes severe metabolic disturbances, problems of fertility, and they all die at a premature age. So not having any adipose tissue is very bad for health, but having too much of it is also a problem. And we know that. We haven't seen this quite a lot. We eat too much, we spend too little of this energy we consume through dietary diets along the days, the years. And so the adipose tissue does what it does best. This is taking all this excess of energy inside its cells, inside the fat cells. This is not a problem. This tissue, this organ has evolved for the last 600 million of years to do exactly that. The problem is that we continue going. We continue taking a lot of energy that goes through our bodies. And this is a reaching point, a peaking point in which our fat cells say not more. This is enough. We have burnout, if you want. And they become stressed. And then the cells that are stressed, they do what they do when there is this kind of problems. They call for help. This is what we know as meta-inflammation. When the metabolic cells are no longer in the charge of the energy effluxes, they call for the help of immune cells. This is some sort of coup d'etat in which the immune cells take over the control of energy fluxes in metabolic tissues, like adipose tissue and also the liver. The guy in this picture is none other than Professor Otamis-Ligel, the winner in 2018 of this ESD Novo Nordisk Foundation Diabetes Prize. This is as much as 100,000 euros to the researcher for its contribution to the advance in the knowledge of diabetes and related disease. And what this guy did to obtain such a recognition in the field of diabetes, Professor Otamis-Ligel published a number of wonderful papers describing how the immune cells infiltrate and activate inside our fat depots, inside our adipose tissue, promoting what we know today as inflammation. This is the basis of this inflammation, the secret killer that they said in Discover in the journal Time. Today we know that the excess of adiposity leads to this inflammatory profile that it's very bad for the metabolic homeostasis. This is just one of many other excellent researchers in the field of obesity, type 2 diabetes, and metabolic disease, lifestyle disease, that have built their research working in the adipose tissue that has increased during the last 70 years to become one of the most important tissues, organs in the control of the energy homeostasis, and so in the prevalence of what we know today as lifestyle disease, that at the end are disease related to the excess of adiposity and aging. How we contribute to this wealth of knowledge about the adipose tissue. I used this graphical abstract from a paper we published in Molecular Metabolism last year that I think it reflects pretty well what we do in our lab. Our starting point is almost always this cohort of human samples, human adipose tissue available in the fat bank. We screen this available sample and looking for some features associated with the obese profile and the risk of disease in this kind of patients. Thanks to our collaborations with the surgeons of this hospital, we get access to fresh human adipose tissue that we can process in order to dissociate, in order to segregate different cells that compose the adipose tissue. This is the mature adipocytes, MA, and the rest of the cells in this fat, the stromal vascular cell fraction. We also perform cultures of progenitor cells, the pre-adipocytes, PA. We sit these cells in plates, and we use a hormonal cocktail to obtain the mature adipocyte, which is the cell we need to study the most in order to comprehend what happens in adipose tissue. Here in the flick, there is in black, there are the lipid droplets that accumulate in these pre-adipocytes, while these cells differentiate into mature adipocytes. With this mature adipocyte, we can perform engineering genetics to knock down or overexpress the genes of interest, the target genes we have identified in human samples by means of silencing RNAs and plasmids, for instance. And finally, we use a number of biochemical techniques, protocols to characterize the result of this modulation. This is connecting obesity to adipocyte dysfunction through the identification and characterization of some target genes, target molecules. This is an example of this procedure I would like to show you. This is a study we published in Journal of Clinical Endocrinology and Metabolism in 2015. The starting point was a cohort of 25 morbid obese patients that underwent bariatric surgery. This is the surgical removal of the stomach to allow significant weight loss. We obtained adipose tissue at the beginning of this surgery and also 2 years after significant weight loss. And we performed transcriptomics in these two time points for the adipose tissue. Like that, we identified more than 5,000 transcripts that changed significantly after weight loss in the adipose tissue, 70 of them with a fold change over 2 in one direction or another. Here, this is the 70 genes that show significant changes after weight loss. Most of them were related to the cell development and inflammatory response. They come from the baseline red, increased expression to the post-weight situation, post-weight loss situation, decreased expression in green. And on the other side, we had these genes related to lipid metabolism coming from the decreased expression in the adipose tissue of obese people and increased expression green to red after significant weight loss. We were surprised back then to find between these 70 target genes a couple of genes that were not expected to behave like that. In fact, they were not expected to be expressed at all in the adipose tissue. The PKP2, plakophilin-2, and the olfactomedin-2. And so we wanted to see what was the meaning of these two genes in our data set. Plakophilin-2 is pretty well known among the cardiologists because mutations affecting this gene, PKP2, are known to be related to arrhythmogenic cardiomyopathies. Patients suffering from this mutation tend to have more risk of sudden death due to a heart that is not that strong. Apparently, this is phenotype relies on the appearance of adipocyte-like phenotype between the cardiomyocytes of people having this mutation in the gene of plakophilin-2. Of course, there was nothing about this gene in adipose tissue. The other gene, olfactomedin-2, we identify in our data set was even less known than PKP2. There were 22 papers when I started to study this olfactomedin-2. Most of this publication was coming from our colleague ophthalmologist in the National Eye Institute, Stanislav Tomarev, Professor Stanislav Tomarev. He is also the owner of the only olfactomedin-2 knockout mice. He was keenly enough to provide this mice model to perform some experiments regarding the metabolism. thanks to this collaboration, we were able to see that this olfactomedin 2 knockout mice, apart from some impaired visual, olfactory, and motor functions, this mice is protected against obesity. This is quite a finding back then. Probably was published in Metabolism in 2022. Apparently, the lack of olfactomedin at the level of brain promotes the thermogenic activity of these mice, and these animals are not able to develop obesity no matter what you do with them. High-fat diet, high-salad diet, they are protected against obesity. Anyway, we were not interested in the expression of these proteins in heart or brain. We were interested in the expression of this plecophyllin 2 and olfactomedin 2 in adipose tissue. And so we continue our research through the validation of these results in studies of pre-post weight loss, here in the expression of plecophyllin 2 in subjects with obesity, the recovery after significant weight loss, associations with the body mass index. The expression of plecophyllin 2 is inverse to the degree of obesity, especially in the subcutaneous fat. The more obese you are, the less the expression of this protein, the gene, in your adipose tissue. PKP2 is also associated with the adipocyte phenotype. There's a clear increase of this PKP2 during the differentiation of fat cells, and also the inflammatory stimuli was able to compromise the expression of this PKP2 in mature adipocytes. More or less the same for olfactomedin 2 is a protein, the expression of which the disease gene increase after weight loss, the inverse association with the body mass index in both fat depots in this case, the association with the obese phenotype. This is also a gene that is highly expressed in mature adipocytes, the difference with other olfactomedins, and also as the same as in PKP2 research line, inflammatory issues can compromise the expression of this gene in mature adipocytes. So we have these data sets of results showing the association of these two genes with the phenotype of adipocytes and the obesity phenotype. We wanted to see what was the functional relevance of this decreased expression of these two genes in human adipocytes. And so we performed first this experiment with silencing small interference RNAs to decrease the expression of olfactomedin and PKP2 during 3 days post-differentiation in human adipocytes. Here, then we perform transcriptomics. Here they are the results for PKP2 at your left hand and olfactomedin 2 at your right hand. There were not many change at the level of transcriptomics, but we found quite a stunning evidence of the decreased expression of genes related to cell cycle, both in the case of the silencing of PKP2 and the silencing of olfactomedin 2. Okay, in blue, the pathways that were related to cell cycle were significantly decreased upon this knockdown of these two genes. This was something kind of surprising because, mature adipocytes is a fully differentiated cell like neurons, and they are not supposed to undergo any cell cycle performance. They do not do proliferation, and that, well, we were kind of disappointed by these first results. And so we decided to perform another experiment of knockdown, the same one, but with a sustained silencing of these two genes by means of lentiviruses. This silencing in mature adipocytes was maintained during 6 days. And we perform again transcriptomics to see what was going on here. There were much more changes upon the silencing of precophyllin 2 at your left hand and also olfactomedin 2 at your right hand. Again, the pathways that were more clearly decreased upon the silencing of these two genes were pathways related to cell cycle. Here in blue, the 3 top decreased pathways were related to this cell cycle. So we had two research lines with an output that was not very clear or very expected. We didn't know what to do with these results at this point. Luckily for us, in 2021, the Swedish school of Peter Arner, Professor Kirsti Spalding, published this Nature Medicine, in which she said that yes, there is a cell cycle, there is an active cell cycle in mature adipocytes as well, and that these cells need this active cell cycle to handle the excess of energy that comes to them in obese people. At the moment, this cell cycle fails to perform its activity, then there is the senescent profile appears in mature adipocytes, and that leads to premature senescence. And you know that senescence is a very bad thing inside the tissues, even worse than inflammation. So, thanks to this paper, we were able to explain and finish our studies. We get to this conclusion through the study of two different proteins, two different genes that were expressed in adipose tissue, PKP2 or factor 2, to conclude more or less in the same line that our colleagues in Sweden. The defective olfactor 2 or PKP2 in mature adipocytes in obese people can impair cell cycle in these cells when inflamed, and that this impairment can drive the senescent profile in mature adipocytes. This is the past. The two papers we published last 2023 and 2024. Factor 2 was published in this year, in 2025. Now I would like to talk a little bit about the studies we are conducting in the few last years. This COBADIS program, Connecting Obesity to Adipocyte Dysfunction, the same title of this presentation, in which we have been working for a while now. We take advantage of the samples. I explained to you before the samples of adipose tissue in obese people before and after the bariatric surgery to perform not transcriptomics, but proteomics and lipidomics. You see, this is the profile of proteomics and lipidomics performed in the adipose tissue of obese people before and after the weight lost. We also take advantage of perform this analysis in the mature adipocytes isolated from a cohort of obese people and also lean controls. In these mature adipocytes, we perform as well proteomics and lipidomics. Finally, in a third study, we wanted to see to what extent inflammation, macrophage-derived inflammation can account for the changes we see in obese adipocytes. And we performed this experiment in vitro, the proteomics and lipidomics of mature adipocytes challenged with the macrophage conditioned media. Through these three studies, we perform, through the proteomics results in these three studies, we saw that there was a kind of clear coincidence at the level of the pivots affected in each study. You know, weight loss in bulk adipose tissue, obesity in adipocytes, and inflammation in adipocytes. The pathways affected, according to our proteomics study, were all related to mitochondrial function, oxidoreductase activity, OXPHOS, and lipid metabolism. We move on with this, the implementation of these three sets of results, looking for coincidences at the level of proteins and also at the level of lipids. After identifying this set of 177 proteins associated with the inverse to the obese phenotype and 156 proteins inverse to the obese phenotype, we use the results of transcriptomics in other data sets to try to confirm the presence at the transcript level, the changes we have find at the proteomics level. And so we shortlist our gene or gene candidates to 86 and 41. Here is what we got at the end of this procedure, trying to implement the results coming from 3 different studies. We got a final amount of 127 proteins or genes, transcripts, that were significantly associated with obesity, weight loss, and inflammation in mature adipocytes. And we wanted to see whether these genes were able to explain the risk of disease in obese people. And so we performed these statistics. I'm not going through these kind of results, linking the expression of these genes in adipose tissue with body mass index, with the levels of HDL, triglycerides, and fasting glucose, and trying to figure out to what extent these 127 features in adipose tissue are able to explain the risk of disease, the risk of metabolic disturbances in obese people. No. After doing our integrative analysis, we discover, or we shortlist our genes to 38 genes that, according to our results, are able to explain the risk of the prevalence of metabolic disturbances in obese people. Beyond the obesity, there are these 38 genes that only in mature adipocytes can explain metabolic impairment. If you pay attention to the graphical abstract at your left hand, you have seen that we also studied the endothelial cells, ECs. You know that endothelial cells represent the 7%, less than the 7% of cells in the adipose tissue, and yet these cells are very important as they provide with oxygen, nutrients, and waste removal. In the case of adipose tissue, which is a highly it's a highly complex tissue and very hydrophilic. So, and the relevance of these cells is key for the well-being of this adipose tissue. Because a matter of time, I'm not going through these results. Hopefully, I will be able to present these results in the future at this society. For the moment, we will stick to mature adipocytes. This is the take-home message coming from this study. How nicely the study of adipocytes in vitro, ex vivo can show us what's going on in bulk adipose tissue. This is to say, much more of the changes we find in adipose tissue when we gain or lose weight is representative of what happens in adipocytes. We also saw how important immune cells are to represent these kind of changes in the adipose tissue of obese people and also after weight loss. Next is the future of my research and the research of the investigators in the field of obesity and related disease. I'm not going to show any results, just what I think it's a correct pipeline to further dig in the pathophysiology of obesity and aging as well through the nature and the biology of this, to me, paradigmatic tissue of very, very important in this kind of disease. For instance, the study not of the bulk adipose tissue, but the different cells that constitute our adipose tissue, they are very important, and they show different patterns depending on our metabolic state. And I think it's very important to study each cell independently. Last week, I was reading a paper published in Nature, in which they show that neutrophils participate in the yo-yo effect. You know, the regain of weight in people that has lost weight. You know, neutrophils in adipose resident neutrophils can explain the yo-yo effect in obese people regaining weight. And also, of course, it's very important to leave behind to some extent the mice models for the study of lifestyle disease. In my experience, the study of mice models is not such well, it's a model, of course, but it shows some problems in the translation, in the cross-species gap. There are other ex vivo invents, such as these mini organs, or that we can study in our lab to figure out what's going on with these different organs and tissues. I think this is a nice objective in the case of adipose tissues. We were able to perform mini organs, mini adipose tissue in our lab and see how they behave under different treatments, how they respond to metformin, or how they respond to different treatments. We will be able to personalize our medicine according to the adipose tissue. If adipose tissue is so important for metabolism, the study of these mini organs of adipose tissue can provide great insights in this respect. We are doing some research in this field. There are many difficulties, as you can expect. It's easy to an extent to study the stromal cell fraction and perform some organoids from this collection of cells. It's much more challenging to study the mature adipocyte. This cell that performs is quite different to the cell we obtain in the lab, and we really need some platforms to study these mature adipocytes ex vivo. This is more or less what I wanted to show to you in this webinar. You know, the excessive body fat is bound to increase risk of disease. It's very important. It's been the most important research topic in the field, as the prevalence of obesity is increasing around the world so much. Adipose tissue is one of the most prominent defenses. And talking about defenses is not a problem. It's a defense against inappropriate lifestyles. If we understand how it works, the cells that compose our adipose tissue, adipocytes, and so on, we can explain. I really think we can explain disease causation. I'm talking about metabolic disease, but also many other diseases associated with the body mass index. For instance, cancer is also a metabolic disease. I would like to finish this presentation thanking to all my co-workers and collaborators that helped me in performing my research during all these years. In this picture, there are people that are very good at proteomics, lipidomics, computational biology, and of course the clinicians. I will extend my thanks to the surgeons that provided the great patients for my research and provide this important sample that is fresh adipose tissue to perform my research. Thank you very much for your attention. If you have any question, I will be glad to answer.
Thank you, Paco, for this very interesting talk. As he said, now the session is open for questions, so you can actually type your question in the chat, and we will translate the question to Paco. There is actually one question already from Connor Perry. He said, he asked whether you have looked at the differences between differentiated adherent mature adipocytes or floating mature adipocytes. Whether there are differences in the way they behave, and what's your opinion on using one or the other model to study adipose tissue biology?
This is something that has, this is a very important question. Thank you very much. It is something that has, you know, it's one of my key objectives in this sense. I want to study the actual mature floating adipocyte that comes from our adipose tissue, because this cell is not the same that the cell we obtain in vitro by differentiating these precursor cells, you know. But it's very challenging, very difficult to study these floating adipocytes, avoiding the differentiation and so on. We are trying many platforms to try to maintain these cells alive and actually study these cells ex vivo, but it's kind of difficult. You may know that they tend to differentiate if they don't find the proper system. Whereas we are looking for that. But of course, at the level of molecular analysis, it's much more important to study the floating adipocyte, this cell that comes from the adipose tissue. The potential, the mirroring capacity of these adipocytes we obtain in vitro, it's limited, unfortunately.
Following this question, actually, how different are those cells? I mean, talking about independent of the origin, I think that you were talking about isolating adipocytes from human tissue and then keeping them floating, and comparing to cells that have been differentiated. But even if you differentiate those cells on a plate, let's say, and then just put them, detach them from the plate, so they are not in contact with the substrates and so on. So how different are they? I mean, is there any study that tell us the degree of difference and why they behave differently?
There are many papers showing, but not many, but quite a lot. I have shown some of these PCA in my presentation that always, the mature adipocyte, isolated mature adipocyte, even if you perform proteomics or transcriptomics, is always, different from the rest. You can do organoids, spheroids, or differentiated adipocytes of actually bulk adipose tissue. They segregate. We have not been able to mimic the exact phenotype of these mature adipocytes isolated from human adipose tissue. And this is, I think, it's a challenge, and it's a very important question we need to tackle in the future. To obtain this mature adipocytes and study these mature adipocytes, I think they will answer many more questions that bulk adipose tissue, spheroids, or whatever you can imagine.
So, and with all this pressure about using alternative methods to the experimentation, I guess you just mentioned the several possibilities to study adipose tissue biology, organoids, organs on a chip. What's the state of art of these methodologies, and how suitable do you think they are for the purposes of research?
It's a good question too. There is a paucity of this bibliography looking for this kind of, there is a lot of organoids bibliography in cancer, brain, kidney, and so on. There are many studies of organoids in many other organs, but not that much in adipose tissue. It seems that there is a challenge to study this mini adipose tissue. But yes, I think it's that there's still work to do, but we are going in this direction. I think we can get to it in the future. Maybe the spheroids, and then we can perform the spheroids from stromovascular cell fractions. This is quite easy, but then we need to differentiate these adipocytes precursor cells to obtain spheroids of adipose tissue. But then again, this is not the real thing, Again, we are using adipocytes that have been created by hormonal components in silico. It's not, it's not what we want. As far as I can tell, the use of scaffolds in which we can embed the adipocytes and then put in also other cells mimicking, containing adipose tissue. I think we can perform this research by using scaffolds, can get the trick done, maybe. But it's too soon to say that any of this. Maybe in the future we can talk a little bit more about this.
There's one thing for me that it's really important. I think that for to get into the metabolic function, it's not just a matter of one kind of cell. Exactly, you just mentioned, and it's really important to have the crosstalk with other cell types. This happens in every organ. So I think that many of us are trying to get there with organ on a chip or trying to do our own trials, but I think that we still have many difficulties in getting into there. So even though we know that we need to little by little get the animal models far away, I think that it's the only way we can see the crosstalk. And in metabolism, this is something that it's fundamental. I mean, like having the whole body. I mean, it's not just a matter of an organ. Even with a cancer, that it's limited to an organ, as you mentioned, it's a metabolic disease. So at the end, also other organs have their input. So I don't really know how can we get all that into an organ on a chip, maybe other stuff, or into I still doing my trials, but I think that we are not there yet.
I'm thinking big science fiction if you want, but I imagine a future in which we can obtain some slightly piece of adipose tissue from the subcutaneous fat of our patients, or the patients of endocrinologists, and use this small piece of adipose tissue to check what's the better treatment for its obesity, its metabolic impairment.
That's what we want. And in cancer, it's exactly the same. It's going towards the personalized medicine. But we need to recreate that. So because then also in organs it's really difficult to mimic the starting condition. So I think that we need we have for a while the animal models, even though we need to get rid of them. But yes, I think that Paco, I really like your presentation. Thank you very much. And I think really interesting stuff there with the two proteins that you found that I don't know if I got this right, but when these two proteins, PKP2 and the OLFM2, those were down or upregulated when they lose weight after the bariatric surgery?
Upregulated.
Upregulated.
After weight loss, yes.
Okay, so if you do the downregulation in your animal models and you get them fat, you get the obesity over there?
Yes. In the case of olfactomin 2, we perform the specific knockdown of this olfactomin 2 in fat depots, and these mice gain more weight.
Gain more weight, and they had a downregulation in E2F targets.
Say again, sorry?
And because one of the things you pointed out was the E2F targets.
Yes.
And you found that they were downregulated, right?
When this two protein fail, this expression of this target.
And while the animals got more obese.
As well, there was a decrease in the expression of this kind of markers, cell cycle.
This is really, yes, because this is one of our lines of research. We work in E2F, E2F transcription factors, mainly E2F1 and E2F2, and we work on metabolic diseases and cancer. And this is really interesting because we know that when we target E2F1 or E2F2, we get thinner, they lose weight, they get relief of all the fat, the liver looks wonderful. I mean, they recover from muscle D and everything. So I'm like, my head is going nuts trying to figure out how these two proteins can somehow downregulate the E2F targets. I don't understand. Have you done more related to this? And do you know how your livers look like?
The livers was unaffected when we regulated this protein in the adipose tissue, but of course was much well in the knockout mice, that's for sure. With regard to the mechanistic, in the case of PKP2 was a relation with PKC. This is related to the transcription of genes, but we don't get to go very deep in the mechanism with regard to olfactomin 2. We are still working on it.
Because if you downregulate the cell cycle, this is something that we and others have done, you get an upregulation in fatty acid oxidation. And even in oxidation of glucose, it's like you get that profile. So
Okay.
that's why I am trying to connect
That's good.
the idea.
Mhm.
Yeah.
Sure.
You have a total remodeling in metabolism because E2F transcription factors are not just cell cycle modulators, but also metabolic regulators.
Okay.
So that's, yeah,
That's very interesting.
very interesting. Yeah. Thank you.
Can check on that.
We don't have questions from the audience. I will encourage the attendants to this webinar to ask questions to Paco. You can do it if you're watching at the seminar through YouTube. You can actually use the chat on YouTube to ask questions to Paco. Meanwhile, I would like to ask about also these models that you've been using on the olfactomedin tool. Well, see, this protein is basically highly expressed actually in the brain.
Yes.
And in the olfactory neurons, if I'm not wrong.
That's correct.
Which part of the phenotype of those mice related to body weight do you think are related to alterations in neural circuits, like volume, energy expenditure,
That's a good question.
Activity?
When it was Miguel Lopez in Galicia who performed this kind of studies with olfactomedin 2 knockout mice, because here in Girona there is not animal facility, and so we have to, that we have to ask for help the researchers that have access to this animal. And I used to say, Miguel Lopez used to say that he was amazed. He has never seen something like that. The mice, the olfactomedin 2 knockout mice was unable to develop any obesity. It was so thermogenic active that no matter what you did with these mice, they did not develop any obesity. They maintained the weight check. This is something coming from the brain. That's almost secure. But there are other deficiencies in these mice. They are impaired for olfactive function, for instance. And when you get there is a link between olfaction and thermogenic activity and the protection against obesity. And so we think that is the mechanism. The decreased olfactive sensitivity in these mice lead to increased thermogenic activity and the protection against weight. With regard to the specific knockdown of olfactomedin 2 at the level of fat depots, but the mechanism is more tricky. But we think that the lack of this protein is impairs the differentiation of adipocytes. And so there is hypertrophy in the remaining major adipocytes inside the depots. And so the increase in adiposity in these mice due to the knockdown of this protein in adipose tissue.
And have you tried to? I mean, I don't know if there exists adipocyte-specific knockout or similar model that essentially allow you to.
Yes, Stanislav Tomarev is telling me that hey, I have the flox mice, olfactomedin 2 mice to perform the specific knockout in adipocytes. You want it? But I cannot. It was very interesting working with these two proteins, but it was exhausting, And at this point, we are moving to other things, and I think the topic is closed for now for us. The topic of olfactomedin 2. Yes, of course, we had a lot of problems because the whole body olfactomedin knockout mice, and so we had to perform the knockdown in adipose tissue by means of lentigraph. And but they are still working. But there are still questions about what will happen if we knock out the gene specifically in adipocytes. So that's a good question. For the moment being, it will remain unanswered. Thank you.
Yeah, Patricia, I don't know if you have any other question. I still have a couple of questions more, but
no, I'm saying
there is
it's also another thing that is so amazing, it's when you do the lipidomics of those patients or humans with obesity, how all the lipids go down, including, because you could think about triglycerides and so on, but also you had sphingomyelins, I could see, and so also the mechanism for that, what would it be? Do you think that because these people lose weight because they don't eat that much, because they have the bariatric surgery, right?
Yeah.
So they less, so they have less fat, and at the end they recover from all those toxic lipids, because the triglycerides are just the ones that we are seeing, but the other ones, or that toxicity.
Overall, there was an increase for most of the lipid species we studied in adipose tissues, like when the adipose tissue recovers from the excess of adiposity and the inflammation, they produce, all the lipid species were increased after weight loss in adipose tissue. And the opposite, in mature adipocytes, when they were inflamed, the production of overall lipid species was decreased, even ceramides and all these kind of lipids. So,
maybe I look backwards.
Yeah, it's more like something general. But it was curious to see that the amount of triglycerides, the kind of triglycerides changed quite significantly. There was an increase in triglycerides, saturated short-chain triglycerides after weight loss, and the opposite in the inflammatory model. It's like, well, that's related to the increase of lipogenic enzymes and factors. You know, the increase the
steroid desaturase and those that include
fatty acid synthase and so on. They all increase, and so the more production of this kind of
yes.
I think there is another question in the chat, I think.
Yeah, there is another question. Yeah, it's again Connor, who's asking what are the differences between different adipose tissue depots in the body? What's difference between cell proportion, cytokine composition?
Yeah,
Can you talk about this?
That's of course another great question. In that study of pre-post weight loss, we are studying subcutaneous adipose tissue because the accessibility of this adipose tissue. During the surgery, we can obtain both subcutaneous and visceral. You know that the visceral adipose tissue, omental adipose tissue, inside behind the muscle wall in the abdomen is the most important at the level of metabolic disturbances. No? The visceral is the one that is supposed to perform the problems at the regard of metabolic disturbances. In the pre-post weight loss, we had access to subcutaneous fat, but our studies in isolated cells, they are all in visceral fat. Because the visceral fat is quite more easy to obtain enough material to perform our analysis. You cannot obtain subcutaneous adipose tissue from lean subjects because there will be a gap, and this is not good. They won't allow this, and so we are focusing in visceral fat because the relevance in metabolic disturbances and so on. But at the end, I think that we can use both to understand the nature of this adipose tissue. At the end, I think we can perform analysis in both the pots of fats to get a fully picture of what's going on in these two depots. They are different, of course. They are very different, but in the molecular basis, they are the same. So we can use both to better understand the pathophysiology of obesity and related disease.
There's no more questions from the audience. I have a very last question. So both of those genes that you talk about, OLFM2 and PKP2, both of them, one of the common features is that they can regulate genes related to the cell cycle.
Yes.
And Which is somehow surprising in the sense that if you're looking at mature adipocytes, right? I got it right. So are those two genes somehow functionally related, or they have nothing in common? It's just a simple coincidence that they both regulate?
For a while, that's a very good question indeed. For a while, I was wondering. We made many experiments, tried to see why they behave the same in all our experiments. You know, there were two lines in parallel because they came from the same starting point, the same data set. No, but no, there is no relation as far as I know. There is no interregulation one against the other. It's just they behave the same. They are important for the biology of adipocytes, and that's it. They are related to the pathophysiology of obesity the same way that other proteins can be related to the pathophysiology of obesity, the biology of adipocytes, the function of adipocytes. But no, they are not related as far as I can tell.
But OLMF2 increases thermogenesis, right? That's what you mentioned.
The impairment of this,
the knockout
of this gene in brain, I think. We think that it was in brain. Is responsible for the protection against obesity in knockout mice. Yeah.
But the adipose tissue, no. So if you knock
in
out
the adipose
tissue,
the knockdown of surfactant 2 promote obesity.
Yeah, that's quite amazing. I mean,
tricky. Yeah, it's tricky. Yes. It's the potency of bibliography regarding surfactant 2 also makes things quite difficult.
But if you have the global knockout, how is it? Fat gains weight or not?
It's protected. The global knockout is protected against obesity. It doesn't develop any adiposity, and we think that's because the thermogenic activity of this.
Exactly. No, that's what I understood at the beginning. That's why I was relating increased thermogenesis, decreased cell cycle. So that was my because that way I understand it. That makes sense for me in my so okay. Well, thank you very much. Yeah.
Thank you to you. Thank you very much.
Yeah.
Okay, so I think that if there are not more questions, I think that we can close the session. I think I'd like to thank Paco for this nice talk and for answering all these questions, and all the guys that attend the webinar for being here. So thank you very much, and I think that we can close the session. Paco, we can stay online and chat later.
Don't leave yet.
Okay.
Thank you. Bye.
Bye.
Ya se ha cerrado el YouTube.
Vale. No, es que yo es que en mi cabeza, nosotros llevamos muchísimos años trabajando